Okay guys, so as promised, I'm doing a video over the macromolecules that we're working on. These will all be covered on your test, so I'm just going to go over them briefly in case you have any holes in your notes that you need to get caught up on. So first things first, what we say macromolecules are or biomolecules are organic molecules. So all living things contain organic molecules, and of course the ones that we're focusing on are carbohydrates, lipids, proteins, and nucleic acids. So the thing about organic molecules is all of them contain a carbon backbone, hydrogen, and oxygen. So I broke up the macromolecules based on the composition of what the carbon, hydrogen, and oxygen that are found. Carbs and lipids are strictly carbon, hydrogen, and oxygen. Protein have some nitrogen, which is inorganic. And nucleic acids have carbon, hydrogen, nitrogen, and phosphorus, which nitrogen and phosphorus would also be inorganic. So macromolecules, all of them, when we're talking about just a generalized term, every one of them, except for lipids, contain a different monomer. Monomer is a small individual molecule that connects to make up the polymer. So it's more or less the subunit, and the polymer is the generalized term that we use for the large molecule that is composed of the repeating individual monomers. And all organic molecules important to life have these large polymers. So we'll start with our first one, and our first one is actually carbohydrates. Carbohydrates, a good example of them would be sugars, starches, cellulose. The sources where we get this is, of course, just sugar. Sugar is a perfect example of just a regular disaccharide, which we'll get into that in a second. Wheat, rice, corn, potatoes, all of the carbohydrates more or less are used by organisms for what we call quick energy, because they're easily broken down and we get that quick energy from them, or that sugar high almost that some people feel. So I give the example of a monomer that we find in our bodies, and plants have it, and a lot of things have it. It's glucose. So glucose is its molecular formula is what we say is C6H12 and O6, and the reason being is because carbon, there are 6 of them in this molecule, 5 of which are in the ring. There are 12 hydrogens, and there are 6 oxygen. And so we say that glucose is a monosaccharide, although I did spell monosaccharide incorrectly there, but we call it a monosaccharide because it is a single sugar, and saccharide always stands for sugar. So if we just have one sugar, we call it a monosaccharide, which is the basic monomer for carbohydrates. So when I say that, of course, I just kind of covered that, the monomer equals the monosaccharide, or the basic building blocks. To make polysaccharides, which is what you see below, a bunch of monosaccharides connected together, we use what we call a condensation reaction, or you could also refer to it as a dehydration. This is a reaction that removes a water molecule, and by removing the water molecule we create a new bond, and that bond is very specific to a covalent bond. Covalent bond that we call, when we're talking about sugars, as a glycosidic linkage, which I have that word right here. So when we refer to the covalent bonds found in sugars or in carbs, we are talking about glycosidic linkages. If I were to break up my polymer and my polysaccharide, break it down and break those glycosidic linkages and return it to its monosaccharide form, I would call that reaction a hydrolysis reaction, because I would be putting water in order to break up the bond. So moving on to lipids. So a good example of lipids are fats and oils. A lot of the sources that we get the lipids from are waxes, steroids, butter, cholesterol, animal fat, different things like that. So organisms use this as long-term energy storage and for insulation. And lipids, of course, do not mix with water, because we say lipids are non-polar. And since water is polar, we say it doesn't mix. So the lipids category out of our macromolecules is a little special because it actually does not have any monomer subunits. They don't have monomers, although Probably the closest thing to what you would consider a monomer to be would be glycerol and the three fatty acids that our fat molecules are made of. And so one polymer that you can get out of lipids is if I combined one glycerol molecule with three fatty acids, as you can see here, we would call this a triglyceride. It is a type of fat, of course, so we find that in our body, but pretty much its units are what I say here: glycerol and a fatty acid, three fatty acid chains. And fatty acid chains are simply just all carbon and hydrogen connected together. So that's more or less kind of what they look like. And again, the triglyceride would be the polymer out of this. So one category of lipids is we have fats, which again you can see here. If I were to look at my triglyceride from its molecular formula standpoint, I will notice that it also contains covalent bonds. And these covalent bonds, when we're talking specifically in lipids, we call them ester linkages. So please don't be confused with what we called the glycosidic linkages in carbohydrates. Those are specific for carbs, and ester linkages are specific for lipids. And so again, like I said, triglyceride is an example of a fat. Fat is one category. There are also two other categories that we have. We have what we call phospholipids, which we'll get to in a second. And then we also have a group called steroids. Steroids is also a type of lipid. So good fats versus bad fats. I know fat is a buzzword that we throw around, and we say that some fat is good, some fat is bad. Bad fats are usually made up of triglycerides and have only what we call saturated fatty acids. Saturated simply means that all of the bonds between the carbon and hydrogen are single bonds. So there are no double bonds in saturated fat, okay? So again, like I said here, they lack double bonds and they are less bulky, usually, which means that they can be packed together more tightly, which makes it harder to break down. So saturated fats can also sometimes cause increased cholesterol within humans. Now, the good fat that we call is unsaturated fatty acids. This is when our triglyceride within it, the fatty acid chain, has double bonds. So, meaning between the carbon and the hydrogen, there are double bonds. And these double bonds make the molecule very bulky, which makes it a little bit easier to break down for us because it's increasing the surface area of the molecule, which allows our different enzymes to go in there and begin to break down. And we get a lot of these unsaturated fats from plants. We get usually saturated from animals, but we get unsaturated from plants. So eating a high diet full of unsaturated fatty acids is actually very good for you. You can get some of these unsaturated fats through like different nuts and things like that. Beans, these are different types of plants that have these fats in them. They will not actually increase your bad cholesterol, which is good. That's why we like to eat them. So, like I said, the next category, we have three, is what we'll call phospholipids out of our lipids. And this is the one that has only one glycerol molecule and two fatty acids. And I kind of have a gross overview picture of what it is here. The also big ticket to take away with phospholipids is they also have a polar head, a phosphate group on top. And this polar head, okay, is what allows it to interact with different water or polar environments. And so it's kind of dual-natured in its polarity. Phospholipids have a polar head, but then again, they also have fatty acids, which we say are non-polar. So those non-polar fatty acids allow them, the phospholipids, to also interact with non-polar environments. So it's a dual nature. And we say that the fatty acids are hydrophobic because they're water-fearing, they're non-polar. And then we say that the phospho head is hydrophilic because it's able to be incorporated in water because it's polar. And so I just wanted to reiterate here about saturated versus non-saturated. If you see kinks, which will equal double bonds in our fatty acid chains, we call that, again, like I said, unsaturated fats. However, if you see it as a straight line, meaning that there are no double bonds, it's just all single bonds, we call that saturated, right? So saturated fats. So I have an example here of what happens when you actually drop a group of phospholipids into like a water environment. If you drop enough of them, they create what is called a micelle. So a micelle is pretty much only phospholipids that arrange themselves so that the inside of the micelle is lined with phospholipid heads, which allow it to interact with water, as well as the outside. However, the one layer that it kind of forms, this bilayer, this double layer, that layer, which you see in yellow over here, is filled with all of the fatty acid chains because they are nonpolar and they are trying desperately to get away from the water or the polar environment. And so it's this reason that these phospholipids, it's really important that they do this, because this is also what is believed to have been the creation of what we call the cell membrane, and how when cells first were created, this is what happened to them. How their membranes were created was because of this phospholipid and its dual polarity. So the next on our biomolecules is what we call nucleic acids. And so a good example of a nucleic acid is DNA or RNA. The sources, of course, are nitrogen, sugars, phosphates. DNA, really, it's, when you think about it, it's just our genetic makeup. It's our coding that is within all of our chromosomes, and which tells us why we look the way that we look. And the monomer, when we're talking about the nucleic acids, are actually called nucleotides. And so I have a picture, a gross overview of what a nucleotide would look like. It's actually composed of 3 components. We have a nitrogenous base, which we'll get into in a minute. It's also composed of a sugar. If we're talking about DNA, that sugar would be deoxyribose, because that's actually what DNA is. Deoxyribose nucleic acid. If we were talking about RNA, the sugar would just be ribose. And of course, the last part is a phosphate group, and so which I have here. So the polymer, of course, is the actual nucleic acid. So we would say that the polymer would be like DNA or RNA. And the actual way that it is composed, we say, is a chain of nucleotides bonded together in an anti-parallel fashion. And so when I mean by anti-parallel, I mean that if you notice that the phosphate group down here is anti-parallel to the phosphate group over there. And you'll kind of notice that the tips of my sugar rings are all pointing one way, while the tips of my other sugar rings are pointing the opposite way. And so this is what I mean by anti-parallel. And you can kind of see, if I were to take all the nucleotides and bond them together, this is kind of what they would look like. And so we're going to talk about the bonding and the way that it works, and what bonds with actually what. When you're looking at a DNA molecule, you'll notice that the phosphate of one group will bond to the sugar of another nucleotide. So it's always phosphate to sugar bonding on the actual backbone of your DNA or your RNA. So here I have, again, the same picture. If I were to zoom in on my, say, nucleic acid, in this case we'll say DNA, this is kind of what it would look like. But the actual bonds that are forming between the nitrogenous bases, which we'll get into in a second, are hydrogen bonds, okay, which you see here. Now, when we're talking about our nitrogenous bases, we have 4. They're broken up into 2 categories. The first 2 are actually what we call purines, and the purines that we have out of our nitrogenous bases are adenine and guanine. I have a little way to remember this that's actually pretty easy. The saying is pure as gold. So if I were to take the first 3 letters of purine and the first letter of adenine and guanine, and I would put it in this sentence, which I have highlighted in red, it's just a fun little way to remember that purines are adenine and guanine. And on the contrast, the other 2, which fall under a pyrimidine category, we say are cytosine and thymine. And again, I have another little saying for that, and it is cut the pie. So if I were to take the first initial of cytosine and thymine, which would be the C and the T, out of my cut the, and then of course pi. It's spelled a little different for pyrimidine, but it sounds the same, so it still works. So again, with the nitrogenous bases, we have adenine, guanine, cytosine, and thymine. And when they bond together via these hydrogen bonds in our nucleic acid, we actually, they bond in a certain way. Only purines bond with pyrimidines, and the way that they do this deals with the amount of bonds that they make. So adenine always binds with thymine, and a good way to also remember that is all of the straight letters always bind with the other straight letters, which A and T are straight letters because they're composed of only straight lines, so that's an easy way to remember. And the curved letters only bind with the other curved letters. So G and C are both curved letters, and they always will bind to each other. So again, back up here where we were talking about the amount of hydrogen bonds it makes. Adenine, when it bonds with thymine, will always make 2 hydrogen bonds. And guanine, when it always binds with cytosine, will make 3 hydrogen bonds. And so that's what you see here. And again, this anti-parallel positioning helps with these specific bond formations. Alright, so the last macromolecule we're going to talk about are proteins. And proteins, a good example of these are meats, nuts, and beans. Of course, the sources are the same, and we use them oftentimes to make muscles, hair, nails, and enzymes. And so when I say an enzyme, I mean a molecule that speeds up or slows down a chemical reaction. Again, the making of our macromolecules and creating those bonds, when we said the hydrolysis reactions and the dehydration reactions, these are all driven by enzymes, okay? And so these enzymes are helping with those reactions. So the monomer of a protein, which of course the protein would be the polymer, we say the monomer are amino acids. There are 20 different amino acids, although you do not have to memorize them, and they are the basic building blocks of our protein. The amino acid structure is what I have below. All amino acids will have 2 sides to it. It will have an amino group, and then it will have a carboxyl group on what we call the alpha carbon. And it's the alpha carbon because it's the one that's in the middle. So it will always have an amino group and a carboxyl group, and then it will always have what we call this R group, which pretty much is a additional kind of molecular structure that makes all of these different 20 amino acids. So it's what makes each one different from the other. That's this R group. So the polymer, usually proteins, are made up of 50 to 500 amino acids, and when they're bonded together, we call the bond between each amino acid a peptide bond, okay, to make what we call a polypeptide, which is pretty much just a bunch of amino acids bonded together. And the order of these amino acids is actually really important when you're dealing with protein and the function of that protein and how it will fold. So proteins, what make proteins special is their shape oftentimes dictates what their function is. So they actually fold into these various shapes to help produce whatever job they're supposed to do. And so we're going to get into the actual folding. So when we're dealing with the folding and the levels of organization, we say that proteins actually have 4 levels of organization. They can have 4, I should say. Most of them only have 3, but we'll talk about it. The first level we say is the primary structure, and this is, again, our polypeptide chain that is bonded together via these peptide linkages, or these peptide bonds. And so this is just, again, our regular chain of amino acids, really nothing too special about it, except that it is the crux of what our protein will be made of, and it dictates what our protein will do. The next structure down is what we call secondary structures, and we have these 2 secondary structures that are usually found. The first one being what we call a beta-pleated sheet. It kind of looks like a crimped sheet, almost like how blinds look crimped. And it is dictated how it gets this crimping or this structure is via hydrogen bonds that happen in between the amino acids. So we can have a beta pleated sheet, or we can have another one called an alpha helice. And again, alpha helice is more like a curly ribbon. And it is also made that way because of hydrogen bond interactions that happen between the amino acids. The next level down is what we call the tertiary structure, and this is the realm that we've gotten in where the folding has all occurred, and we've reached a functional protein at this level. And so we say that our tertiary structure can have lots of different bonds. It can have nonpolar bonds, or what we call van der Waals forces. It can have polar covalent bonds. It can have hydrogen bonds. It can also have these things called disulfide bridges. And all of these bonds contribute to holding together the tertiary structure of our protein so that it can be functional. And our last category, and not all proteins do this, but some of them do, are called our quaternary structure. And this is what happens when a bunch of tertiary proteins get together and can make a common usage or a common function, and can kind of bond together to make this one larger protein that does a specific function. And so each shading on our quaternary structure represents one tertiary structure of a protein, and you can see here I have 4 different tertiary structures bonded together to make this one quaternary protein structure. So some of the proteins that we find in humans, you do not have to know all of these, but I'm just kind of giving you an overview. We have insulin. Of course, insulin helps with the breaking down of sugar when we intake it. We have pepsin, which is actually in our stomach. It helps break down some proteins. There's hemoglobin. Hemoglobin, of course, helps transport oxygen in our blood. And so there's lots of different proteins that humans make. So summing up all of our, what we call our macromolecules, we have a relationship that occurs between them. And so my relationship that I've given here is the nucleic acids help direct the cell to produce proteins. The carbs and the lipids provide the energy that the cell needs to make the proteins, and the proteins are what help determine our physical traits. They can also be the enzymes that can later help break up our carbs and our lipids when we ingest them. So there's a very cyclical relationship that happens with our biomolecules.